Hello friends, welcome back to allornonelaw. This is a medical video lecture, microbiology, microbiology. And today's topic of discussion is Mycoplasma pneumoniae. Mycoplasma pneumoniae. So before starting a discussion on this, guys, I would like to tell you about our channel. Our channel name is allornonelaw, and we have currently more than 600 medical video lectures on different medical topics that are important for USMLE Step 1 to USMLE Step 3 examinations. So guys, please do not forget to subscribe to our channel and try your level best to share our videos and give us a feedback. Okay, we need your love, we need your support. Okay guys, so let me quickly start a discussion on this, and this is really very important bacteria for USMLE examination, not only for USMLE, for other medical board examinations also. So let's start with this Mycoplasma pneumoniae. Okay, so before, I would like to tell you the family Mycoplasmataceae. Okay, so what are the characteristics you should remember about this family? This is a really very important. Okay, so whenever you hear about the Mycoplasmataceae or Mycoplasma pneumoniae, remember one thing: they don't have the cell wall. No cell wall, really very important. No cell wall. Okay, this is a really very important point you should know about. Okay, and they have what you call cholesterol in their membrane. Cholesterol in their membrane. Okay, in their membrane. Okay, so these are the really very important points. And the other point is, if they have the cholesterol in their membrane, means they require cholesterol for the synthesis of their membrane. So if you're choosing any media to grow the what you call Mycoplasma pneumoniae, means that media should contain cholesterol. Okay, right? Yes, good. Now the other important point is they give the what you call fried egg colonies. Okay, on which media? Eaton's media. Okay, all, but remember, only what you call Mycoplasma pneumonia doesn't give, but the other bacteria of what you call Mycoplasmataceae, like Uroplasma urelyticum, right? So Eaton's media. Okay, for Mycoplasma pneumonia, you can think of mulberry shaped colonies. For Mycoplasma pneumonia, it gives mulberry shaped colonies on sterol containing media. As I said, because they contain what you call cholesterol in their what you call membrane, so they require sterol or cholesterol for the synthesis, for their growth. So Mycoplasma pneumoniae gives mulberry shaped colonies on sterol media, but other bacteria of this family gives fried egg colonies on Eaton's media. Okay, so these are the important features of what you call Mycoplasma pneumoniae. So let's talk about this Mycoplasma. They are very smallest free living bacteria. Remember, they are extracellular smallest living bacteria. Okay, so who is the reservoir? Reservoir is human respiratory tract. Remember, human respiratory tract. So what's the mode of transmission? Transmission is but droplets, respiratory droplets, right? So if they're close contacts, then it can spread easily, like in a military, in a school, in a families, okay, and developed countries, okay, or developing countries where there's a crowded population, okay. So let's talk about the pathogenesis. Okay, the pathogenesis, this it goes, and if this is a respiratory tract, okay, uh, let me draw a line over here. So it goes and attaches to the receptors known as the P1 receptors, okay, P1 receptors. There are P1 receptors, that the P1 proteins, okay, the epithelium, respiratory epithelium, there's a P1 here, you can say. It goes and inhibits what you call ciliary action. It goes and then inhibits what you call ciliary action of cilia. Ciliary action is inhibited, okay. So now what happens when the ciliary function is inhibited? There are more chances of what you call progress of the infection. So usually the infection grows up and the patient develops a pneumonia that is known as a walking pneumonia. Not only it acts by attaching itself to the P1 receptor and inhibiting the ciliary action, the other type of action it does is it produces radicals, that is a superoxide radical, superoxide radical superoxide radicals. Then it produces H2O2, there's a hydrogen peroxide, and catalytic enzymes, okay, and catalytic enzymes. These enzymes are, and oxides, or H2O2, hydrogen peroxide, are very dangerous, very lethal, right? So they go and damages the respiratory epithelium. That leads to the what you call necrosis, and the patient will develop a pneumonia, and that pneumonia is known as walking pneumonia. Remember, whenever they give what you call Mycoplasma pneumoniae as an organism, so think of walking pneumonia, okay. So the other, it also, the Mycoplasma pneumoniae produces the interleukins, okay. It acts as a super antigen and produces interleukins IL-1, remember this is really very important, IL-6 and TNF alpha, okay, IL-1, IL-6 and TNF alpha. So you know better how they act, and what is the next thing would be happening, okay guys. So remember about the Mycoplasma pneumoniae, they go and attach to the P1 protein, okay. They inhibit the ciliary action and they produces superoxide radicals, hydrogen peroxide, catalytic enzymes. They go and damages the respiratory epithelium, leading to the necrosis, and ultimately leads to the what you call walking pneumonia, okay. So these are the features. So diseases, you know very well what are the diseases it can cause. It can cause pharyngitis, it can cause a pneumonia, as we discussed, okay. So let's talk about a diagnosis. How would you diagnose this Mycoplasma pneumoniae? Look at this is clinically based, okay. So you can diagnose it clinically, and as I said before, the mulberry shaped colonies on sterile containing media, you can see this after 10 days. Remember, 10 days, not within a 2 or 3 days, 72 hours, no, it's after 10 days. Remember, this is really very important point, okay. Now ELISA, immunofluorescence, sensitive and specific you can do, and the important point over here is positive cold agglutinin, gold, not cold, cold, chill, okay, cold agglutinins, okay. So it's nothing but autoantibodies to red blood cells, remember, and this is not specific, okay. Positive cold agglutinin test you have to remember for Mycoplasma pneumoniae. So now what's the treatment? Treatment is only, you can remember as what you call ECA, there's nothing but erythromycin, okay. Then we have clarithromycin or azithromycin. So mycin's gonna work for this, okay. So thank you so much for watching this video. I'm sure you got some information about the Mycoplasma pneumoniae and the Mycoplasmaticea. In next video, I will be discussing about the urelyticum. This is also an important topic for USMLE Step 1 examination. Guys, thank you so much for watching this video. I'm sure this video was really helpful. Thank you so much. Please do share, please do subscribe, and thanks for supporting us. Take care.